Hello everyone. I welcome you at Advanced Multiple YouTube channel. Advanced Multiple Incorporation actually a knowledge processing and information management organization that is located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir. I'm working as scientific executive with Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Okay, well, today I'm going to share my information, my knowledge, and my experience under the title of Validation and Qualification of Pharmaceutical Process and Production. For this purpose, I collected the scientific information and the research data from different open source like PubMed, Medline, Scopus, Google Scholar, National Library of Medicine, MBAS, Directory of Open Access Journal, ProQuest, CNKI, that is China-based database company, stand for China National Knowledge Infrastructure. Some information collected from Crossref and ResearchGate. Okay, well, before moving forward, I would like to talk a little bit about pharmaceutical validation. Validation is documented evidence that provide a high degree of assurance that a specific process will consistently produce a product that meet its determined specification and quality attributes. Pharmaceutical validation is also important in manufacturing process to ensure product consistency and safety. It involves regulation of all raw material, production procedure, and testing of the finished product. The general rule of thumb is to follow the good manufacturing practice. Good GMP or good manufacturing practice is available all different kind of dosage form, like for injectable, for the suspension, for the syrup, for the tablet. Each dosage form have its own GMP, and there are the three main process or three main steps involved in process of validation flow chart. The first one is the validation of process design. Second one is the qualification of the process, and the last one, the third step or this third phase of validation is the validation continue process. So these are the three steps or three phases involved in pharmaceutical validation. Okay, well, this is actually flowchart that highlight or demonstrate the process of validation of pharmaceutical. There are different steps, different people, different entity involved to validate the pharmaceuticals. There are the involvement of the validation team, testing team, then reviewing board, approval team, all different people involved in pharmaceutical validation. They review, they check the test specification, they accept or decline the criteria to for compliance. They also send it back for the review. So this is the whole process by which a validation of pharmaceutical is conducted in the pharmaceutical industry. Okay, well, there are the 11 elements or the 11 component of validation or qualification: designing, installation, operation, and performance qualification. Fifth one is the risk analysis, then process validation, then computer validation, cleaning validation, method validation, validation of sterility, and biological indicators. All these steps or all these 11 components were involved in the pharmaceutical validation. Now we are going to discuss one by one all of these 11 components. Designing qualification is the documented evidence that the design aspect of the pharmaceutical unit have been checked and approved. Installation qualification should be written evidence that all parts of the equipment are installed according to the equipment supplier and the purchase specification. Operational qualification is actually the documented evidence needed to prove that all parts of the plant and equipment work within their specification and process parameters are within the acceptance criteria. Fourth one is the performance qualification, that is the documented evidence that all parts of the plant and equipments validated to make products of the specified quality under condition of normal production for a longer period of time. Fifth one is the risk analysis. Risk analysis actually prove the possibility of the risk if these are acceptable for the quality of the production. Sixth one is the process validation. Process validation ensure and provide documented evidence that process within their specification parameters are capable of repeatedly and reliably producing a finished product of predetermined quality. Computer validation provides the documented evidence that process controlled by the computerized system are checked and produced and specified product quality repeatedly and reliably. Okay, well, cleaning validation actually is the process, the written evidence that specify the clinic procedure leading to reliable and repeatable results in the cleaning of surface with and without contact with the product. Criteria to fulfill: the concentration of the active substance on product contact surface will not exceed specified limit. Then number of the germs on product surface should not exceed specified limit. Third one is the concentration of other pharmacological active substance will not exceed the specific limit. Okay, well, ninth one is the method validation. That is the documented evidence that give the assurance that the product tested by the method will produce same result. The 10th component of the pharmaceutical validation is the validation of the sterility, biological indicator, then electric method, probe system. The complete absence of viable microorganism for the product is known to as sterility. The process by which microorganisms are removed from the product is called as sterilization. There are different sterilization methods involved in the pharmaceutical industry. Most of the methods used are steam sterilization, dry heat sterilization, gas sterilization, sterilization by the ionizing radiation, and sterilization by the filtration. These are the methods used in pharmaceutical industry all over the world. Biological indicators. Biological indicators are characterized preparation of a specific microorganism that provide a defined and stable resistant to a specific sterilization process. The type of biological indicators are three. The first one is the spore that are added to a carrier. Second one is spore suspension. Third one is the self-contained indicators, and most of the time these self-contained indicators having the dye, and that change the color. With the change of the color, the scientist or the researchers or the technician, they can identify the contamination or indication of some biological degradation. In conclusion, validation process established documentary evidence demonstrating that entire activity carried out in testing and then production at required compliance at all stages. Validation is required regulating agencies and good manufacturing practice guidelines. So data should confirm the process met the condition established in protocol. The documented justification for the approval of the process and release of the lot or batches of the product or the production for clinical utilization should comply with the validation and qualification requirements. That will potentially help to maintain the consistent quality of the pharmaceutical production. Okay, well, I would like to greet and salute to my friends, my colleague, my coworker who have helped and support me to make this video today. Shady Bachkat, Wolfgang, Daniel, Dustin Hacker, then Marwa, Robert, Hudza, Noreen, Amin, Arslan, Ishtiaq, Faisal, and Terry Mill. These people, I appreciate. You are my pride. You are my colleague. I also acknowledge the contribution and help of Advanced Multiple Incorporation and Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Thank you very much for watching the video and staying with me. See you soon in next video. Take care. Bye now.